Multiplexing immunohistochemistry offers an unmatched insight into spatial proteomics in a variety of cell populations and bridges the gap between high-throughput and single-cell technologies. Immunohistochemistry, IHC, represents a powerful tool used for the identification of target proteins in the context of the cell or tissue analyzed. This type of in situ analysis is effective in assessing the localization of proteins in the cell, comparing the expression of a protein in a heterogeneous cell population, and can be used to identify subpopulations of cells based on the expression of specific markers. Traditional IHC methods present significant technical challenges for studying complex and heterogeneous tissues, highlighting the need for more advanced multiplexing approaches to better characterize the spatial organization of proteins and cell type. Multiplexing IHC, which allows the simultaneous detection of multiple markers on a single tissue section, has been introduced and adopted in both research and clinical settings in response to increased demand for improved techniques. A number of highly multiplexed tissue imaging technologies have also emerged, permitting comprehensive studies of cell composition, functional state, and cell-cell interactions which suggest improved diagnostic benefit. These novel imaging techniques are based on cyclic immunofluorescence, tyramide-based multiplexing IHC, epitope-targeted mass spectrometry, or RNA detection. Such techniques provide a comprehensive view of marker distribution and tissue composition, and are poised to solve major questions surrounding the pathogenesis of various complex disorders. In terms of the more advanced, higher-level applications of multiplexing IHC, there are various approaches relying on distinct principles. These can be classified into 5 classes: stain removal, fluorophore inactivation, multiplexed signal amplification, DNA barcoding, and mass cytometry. Stain removal technologies. One of the most commonly used approaches of multiplexing IHC is stain removal. Stain removal technologies, sometimes called dye-cycling, refer to a class of protocols that rely on a cycle of antibody staining, image capture, removal of stain, and restaining to greatly increase the number of markers that can be tested in a single experiment. Here are different examples of stain removal technologies. Fluorophore inactivation technologies. The principle of this technology is roughly similar to stain removal technologies. However, it does not rely on using different pH conditions, denaturation, or photobleaching to remove a stain, but on chemical inactivation to eliminate the fluorophore. Multiplexed signal amplification. One of the problems with detecting multiple markers in a sample is that sometimes the epitopes are present at a low copy number. Multiplexed signal amplification methods aim to address this need, providing strong signals for imaging even small amounts of protein. Here, the tyramide signal amplification, TSA, is a commonly used one. DNA barcoding technologies. Another approach to multiplexing is using the innate properties and specificities of DNA itself. These technologies take advantage of DNA's sequence-specific binding properties to generate highly specific staining, which is intrinsically applicable to multiplexing and mapping. Mass cytometry. Mass cytometry is another example of a non-microscopy-based method of quantifying multiple biomarkers that allows users to carry out multiplexing. Based on mass cytometry technology, several methods have been developed such as imaging mass cytometry, IMC, and multiplexed ion beam imaging, MIBI. I hope this helps. Let me know if you need any more assistance.